The eLife Podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.
Hello, welcome to episode 102 and our first in 2026 of the eLife Podcast. I'm Chris Smith from The Naked Scientist, and this month, how moths can hear plants, and yes, you did hear that correctly. Scientists use trackers to work out how people pick up diseases, and the first look inside a finger in real time to see what fingerprints are actually for. Up first though, to the land down under, and the kangaroo. Now kangaroos alter their postures as they hop faster to store more elastic energy in their Achilles tendons and improve the efficiency of their locomotion. In fact, as they speed up, they burn off almost no extra calories. And now we know how they're doing it. Christopher Clementi is at the University of the Sunshine Coast.
Back in the 1970s and 80s, a group of scientists at Harvard University actually discovered this unique phenomenon that we see in kangaroos. They managed to get these kangaroos to hop on a treadmill while wearing a metabolic mask so they could measure how much energy that they were using. What they noticed was that as they hopped faster, the kangaroos didn't use any more energy. Now, the reason that is so interesting is because if I was to run on the treadmill and as I go faster, I would generally use more energy. It's like, when we're driving a car, we put our foot on the accelerator, we use more petrol to go faster. That's true of most animals except for the kangaroos.
And we knew they were doing it, but we didn't know how.
That's right. So what we wanted to try and figure out was what's changing in kangaroos? What are they doing that allows them to decouple energy use from speed? So we actually used a little bit of sports science technology. So you might see sometimes when sports players are running along and they have, these segments and angles and high-speed cameras all used to figure out what their skeletons are doing. So we had little markers that we would put on these kangaroos and we would get them to hop over force plates. And what that allowed us to do is build a computer model of their skeleton as they're hopping along and measure the forces that they're impacting the ground with.
And at different speeds, presumably.
And at different speeds, yeah. And this was the fascinating thing. When we looked at their posture, we noticed something was changing. As these kangaroos are hopping faster, they're starting to become more crouched. And this is our first clue as to what they're doing.
When you say more crouched, which joints specifically are they crouching?
Their foot design is a little bit different to ours. They have a very long foot, and their ankle joint is the main joint that they use. And as they're moving, they're flexing this ankle joint and they're using that to push themselves forward.
So as they go faster, they flex towards the floor more, that ankle joint. How does that translate then into improved efficiency of movement around that joint, which is what this must be? If they're not using more energy, they're not getting a free lunch because physics doesn't do free lunches. So they must be being more efficient with what they're using. So how are they doing that?
That's right. So the way we normally move a joint is we have muscles, right? And our muscles shorten, and as they contract, they pull on that joint, and that joint moves about the axis. Now, the muscle is not the only thing. Kangaroos have ridiculously long Achilles tendon, and it has rather interesting properties. So when we shorten a muscle, we use up energy to do so. But tendons are like an elastic band. If you stretch an elastic band, it takes energy to stretch it. But when you relax it, the elastic band pulls back together by itself. So it's capable of storing energy. And when we let it go, it releases that energy back. So these two things are happening, and both of those are clues as to what's actually going on in these kangaroos.
How does the postural change alter the behaviour of the tendon then?
What we think is going on is as these kangaroos are hopping along, say at slow speeds, Most of the time, because they're hopping relatively slow, that tendon is stretching a little bit, but not much. And so they have to contract their muscle in order to rotate their ankle joint. But as they hop faster and faster, they will want to try and do less and less muscle work. And the way they can do this is by stretching the tendon more and more. And the trick that they use to stretch this tendon more and more is by becoming more and more crouched, forcing the tendon to be stretched further with each hop. Now, when you're stretching the tendon and not the muscle, that means you can tend to store a lot more energy in these tendons. And that meant that the leftover bit, the amount that the muscles needed to do to push them forward, actually doesn't change as they go faster.
Why is it that just kangaroos have hit onto this ingenious solution? Why can't I do this?
Yeah, why can't we do this? That's a great question. Why wouldn't all animals do this if it's such a neat strategy? There is a cost, of course, to doing this. So when I say we're stretching the tendon more and more, what that means is we're putting it under more and more stress. But the more stress you put something under, the closer it is to its breaking point. So we imagine that these particularly large kangaroos, when they're hopping very, very quick, are probably getting close to the limits of what that tendon can handle before it breaks. And so we think most animals are probably a little bit more safety biased in that they don't want to use this method because they don't want to risk rupturing those tendons.
Does this therefore apply a size constraint? So it's good for animals in a certain range of sizes because they can get away with really cooking things close to that safety margin. But once you add more body mass, biology just can't cope with the load you would be asking that tendon to deliver.
Yeah, exactly. So there's going to be two real the cons, I guess, with this approach. The first one is you probably can't accelerate very fast too, because if you try to, apply too much load too quickly, you're going to snap these tendons. And the second is exactly what you were saying. If we put too much weight onto these tendons, then we're going to get to a point where that safety factor is too low and we're really going to rupture these tendons. And that kind of fits with what we see in nature. There are no giant horse-sized kangaroos. Instead, once animals get really big, they have to just switch from hopping to another type of gait, to something like walking. And so maybe what we see in kangaroos is something like the upper limit of locomotion. And these kangaroos are choosing a type of gait that maybe lets them be very efficient, but it's at the cost of this ability to be very big or maybe very maneuverable.
Christopher Clementi there. When plants dry out, they begin to emit high-frequency clicking sounds, possibly through the process of cavitation, as tiny bubbles pop into existence and then collapse again inside the vascular system of the plant. And now, Yossi Yuvel at Tel Aviv University has shown that some insect species, in this case moths, appear to be able to hear these sounds and use them to locate plants on which to lay eggs. It is the first time, he thinks, that anyone has documented a plant-animal interaction like this.
A few years ago, we discovered that plants actually emit ultrasonic sounds we humans cannot hear, but other animals can when they are dehydrated. And thus, we were interested in examining whether any animal actually uses this sound. And the first candidate are female moths, because we knew that they actually can hear these sounds, at least some of them.
Before we talk about the moths, tell us more about these sounds then. So what plants make them? When do they make them? Does the plant have to be dead to be making them? Or is there some physiological reason why? Or is it just a product of a drying out plant?
We actually don't exactly know the answer yet. I can only say that every plant that we've recorded, and this means many species already, produces these sounds. So my guess is that all of them produce it. probably a physical artifact of being dry. One of the hypotheses is that a process called cavitation. These are small air bubbles generated within the plant's water vessels when it is dehydrated, for example, and they might sound like small pops, which would produce ultrasonic sounds. So that would be one of the hypotheses of how these sounds are produced.
In other words, the sounds are a series of clicks and pops then.
Exactly. And what I didn't say is that they're actually constantly produced. So when a plant is hydrated, it still produces them, but they are very rare, maybe one every few minutes. And once it becomes dehydrated or it's under an attack of some pathogen, it will start producing more sounds. So obviously, this means that there's information. If you're an insect or some other organism that can hear these sounds, you can use this information to know what's the status of the plant, to some extent at least.
If they're ultrasonic, how high is the frequency and what sorts of insects might be sensitive to those sorts of sound regimes?
The sounds that we recorded are around 50 kilohertz with some energy even above that. And actually, we found that it's plant specific. So different species of plants have slightly different sounds. There are different organisms in the world that can hear this. Bats, for example, can hear such high frequencies. Many rodents can, many small mammals, and some insects. Among them, many families of moths can hear ultrasonic signals. Some people hypothesize that this is related to their ability to hear bats. So bats prey on moths, of course, and thus it would be beneficial for a moth to hear a bat and to evade it. And some moths have evolved these very special, what we call tympanic ears. So it's like a small eardrum on their abdomen. And this allows them to hear these high frequencies, which we cannot hear.
How then did you try and probe whether the insects, these moths specifically, were tuning into the plant sounds?
We decided to focus on the egg-laying procedure or process of moths. So once a moth mates, a female moth, she will search for places to lay her eggs. So what we decided is to go for a pure acoustic. That was the first experiment, a pure acoustic signal. So we offered the moth two different chambers without any plant inside, one with the sound of dehydrated plant, so these ultrasonic clicks that I've mentioned earlier, and the other chamber was silent. And we found that indeed the moth prefers significantly to lay its eggs more often on the side where there is a playback of dehydrated plant sounds.
Why do you think a moth would do that? What would be the attraction of a dry plant to a moth to lay eggs?
Right. So this was actually a bit counterintuitive for us, right? Because we know that a moth prefers to lay its eggs on a non-dehydrated plant. So it was a bit of a surprise that the moth laid its eggs on the clicking side. But what we found is that if we add a plant into the system, then the preference changes. So the moth will now prefer to lay its eggs on the non-clicking plant, so on the non-dehydrated plant. So what we think is that maybe, and this is only an hypothesis, maybe when there is no plant around, so there's no odor of a plant, no sight of a plant, then this clicking sound, maybe that is the only signal that tells the moth, and these moths are active during the night, so maybe they often rely on sound more than on vision. Maybe this tells the moth that there is a plant actually. So the sound is the only relevant signal for a plant, and maybe that is what's driving the preference. But this is an hypothesis that we still have to check.
In essence, then, it's sort of any port in a storm. Given no choice, the moth says, well, I'll have to go for the click because there doesn't seem to be any other plant option. As soon as you offer other cues, which would be there from a non-clicking, non-dehydrated plant, there's going to be better food for the caterpillars, the moth picks on that instead.
Yeah, and I just want to emphasize that as much as I know, this is the first time that anyone shows an acoustic interaction between a plant and an animal.
Is it possible to deafen a moth so that you could prove it is the sound that's doing this? So you could wipe out the ability of the moth to hear this, in inverted commas, and see if that stops the effect?
Yeah, so we've actually done that. It's quite simple. So as I mentioned earlier, their ear is a kind of an eardrum. We call it a tympanic ear, and you can simply take a needle and puncture it, and that destroys the moth's hearing. When you do that, then the preference disappears, and then there is no preference of laying it. The moth has no preference of laying its eggs on the clicking or on the silent side.
Any evidence that this might be influencing predators too? in the sense that we know that moths and their caterpillars might be juicy prey for some things to come and eat. Any evidence that things are using these clicks in order to exploit things and find prey?
Right. So this is, of course, one of the most interesting hypotheses we would love to test. So if there's a potential student listening to this, you're welcome to approach us and maybe work on this project. Yeah. So there are many interesting hypotheses. For example, one could think that maybe the caterpillar eating the leaf somehow increases this emission of these clicks, and maybe some predators such as bats can pinpoint these cues and, just as you suggested, eat them. But these are things that still have to be tested. So this is kind of the first evidence for an acoustic interaction, and now there are many, many new hypotheses that come to mind and should be tested in the future.
Absolutely fascinating. So if you fancy that for a PhD or a postdoc, do drop Yossi Yavel a line at Tel Aviv University. This is the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. Still to come, what's the point of fingerprints and the 72 women going up on the outside of the Eiffel Tower? But first, human behaviour is often what lies at the root of infectious disease transmission, and that includes illnesses that we pick up from the environment, like leptospirosis, also known as Weil's disease, a bacterial infection spread by contact with rat and mouse urine. If we can pinpoint what people do and where they tend to go when they catch illnesses like this, then we can mount much more effective public health interventions to cut down the risks. And this is what Pablo Ruiz Cuenca at Lancaster University has taken the initial steps towards doing in one part of Brazil.
What we were trying to find out was how people were moving through their neighborhoods, and if the way they were moving through was maybe affecting their risk for becoming exposed to a specific infectious disease. This infectious disease is called leptospirosis, and it's essentially a bacteria that we can catch from being exposed to rat urine in the environment.
Obviously a very big cause of morbidity and mortality around the world, isn't it? Obviously more common in poorer countries. Where did you focus the study on?
Yeah, so we focused our study in Brazil, specifically in the city of Salvador, which is located in the northeast region of Brazil. These are communities that are quite vulnerable, so they don't get as many resources from local government. People who live there are generally considered more vulnerable.
And how do people in that sort of environment usually pick up leptospirosis, also known as Weil's disease? How do they get it? What do you think the route of exposure is normally?
We know that infection occurs after being exposed to rat urine. We don't really know where this exposure is happening. So it could be happening in their homes. The other possibility is that they're getting exposed in their neighborhoods. So when rats are out on the streets and they contaminate that environment.
And how did you try and disentangle which of the two, or indeed both of those, were at play?
This was something that we were really interested in, but with this specific paper, we didn't really get to the bottom of it. What we did do with this research was we accounted for where people were living. We had their household location, and then we could say, okay, these individuals live in this specific point, and then we looked at how they moved outside of that location. So we tracked their movements through their neighborhoods, and we could compare how they were moving through their neighborhoods.
And what you were then able to correlate movement with outcome, which is do they or do they not succumb at some point to leptospirosis infection? So you can sort of look at what's associated with what must amount to the greatest exposure.
Exactly, yeah. So one thing to point out here is that the way we were measuring our outcome, we were looking at the antibodies that a person has. So this is essentially the protection an individual might develop after being exposed to this disease. The technicality with this measure is that it doesn't necessarily mean that they've been recently exposed. It proves a good tool to understand how movement could affect infection risk, but ultimately it's not a perfect measurement.
How did you track the measurements then? What were you doing to elicit where the people went?
We were working really closely with a group in these communities called the Grupo Impulsor Comunitario. They helped us get people to wear essentially GPS loggers. These are small trackers that people can wear in a pocket or around their neck. It records the location of the person who's wearing the tracker at a set time. So every 35 seconds, that device was recording the location of that person that was wearing it. And we asked these people to wear these trackers for 48 hours. So we could see how they moved in these 2-day periods.
And what emerges then? Are there any patterns that are the giveaways that say, hey, look, there's a really standout high infection risk in this particular group who do this or go to these sorts of places? Did anything jump out?
We found two very interesting results. The first one was that in people who had antibodies for leptospirosis, so people who had been exposed to the disease at some point, we were expecting them to interact more closely with specific risky environments in their neighborhoods. For example, open sewer points and the local river, which was contaminated. However, what we found with these group of people is that they were actually avoiding the open sewer points, which were one of the highest risk locations. And the other interesting result was that women in general tend to move more closely to the stream that was running through these communities. We don't have very clear explanations as to why these two results appeared. You could immediately assume that maybe they were using the stream to wash clothes or wash things. We know that they don't use that water for that. We know that people in general, they understand that the stream is contaminated and they don't use the water. What we don't know is whether maybe they're socializing more around the stream for some reason. So whether, for example, if men are socializing more near their homes and then women are socializing more in more open areas, that's a possibility that we're thinking of exploring.
The end goal of doing something like this is better public health information, isn't it? So that we can guide people.
Exactly.
These are the risk factors. These seem to be the behaviours that place you at greatest risk. This is what to do differently. Similar sorts of things have been done for dengue and providing breeding grounds for mosquitoes, haven't they? So what are the learning points from this? How can you take this now and say, right, okay, we've got some initial findings, but this is what we need to do next in order to really crystallise those public health messages, to really give people concrete advice. This is what you need to do.
The next steps would be to really focus on that question we started with. So trying to understand where infection is happening. So with this paper, we've presented a way to measure how people are moving through their neighborhoods and linking that to their infection risk. And if we can use that with another tool to measure how people are becoming exposed at home, then we can try and understand where that exposure is happening most. The other step is to understand, is there something in how the neighborhood looks like or specific things that are present in the neighborhood that are making people either avoid specific areas or want to be going towards these areas more. And the whole idea behind this would be to use that information, as you said, to try and get people to avoid these exposures and essentially avoid infection.
Pablo Ruiz Cuenca there. Humans, and just a handful of other species, have fingerprints. But what purposes do these whorls and circles really serve? Historically, scientists have speculated that they might improve grip in wet conditions and that the system of ridges almost certainly assists our sense of touch. But no one could actually prove that, at least in vivo. That is, until now, because Giulia Corniani at Harvard has been using a technique called OCT, optical coherence tomography, to see inside the skin in real time as a fingertip slides across a transparent plastic surface. She's been able to document how the forces are conveyed into the skin and deform nerve endings to produce the sensations that they do when we reach out and touch a surface or delve into a pocket for a key or a coin.
At the basic level, we wanted to answer a question: what physically happens inside your fingerprint when you touch something? We want to understand how our skin deform and how these mechanical stimulation has an impact on the subsequent information encoding that is happening in the nervous system.
So, how did you actually approach getting underneath this?
It's difficult to look at what's below the skin surface in vivo conditions. And so most of the study existing in the literature just look at what's happening on the skin surface, or they like do biopsy of small skin pieces and look at them with the microscopes. We wanted to see what's happening during a real tactile interaction because the skin elasticity and the skin properties really change when we just took a piece of skin out of the hand. And so we found this imaging technique that allowed us to look into what's happening below the skin surfaces. And we built this experimental setup to take images in vivo during a real tactile interaction.
What is that approach then, and how does the experiment work? Talk us through it.
So basically, we use this imaging technique, which is called optical coherence tomography, which is something like ultrasound, but that uses lights. And we had some plastic transparent surfaces that we were sliding over the skin surface. And we were using this optical coherence tomography to image what was happening below the skin surface during the interaction between these plastic surfaces and the tip of the fingerprint.
How deeply into the skin can you image with this?
So with these images, we can see for about like 0.5 millimeter inside the skin, but in this like tiny depth, there is a lot of things happening. So basically the skin has a layered structure and there are different layers that have different physical properties. And the first layer is called stratum corneum, and which is basically the protection of our skin. And below the stratum corneum, there is the viable epidermis. And just below the viable epidermis is where the tactile receptor sits. And so we were really interested to see how the mechanical deformation happening at the skin surface propagate to the viable epidermis to see how the tactile receptors are receiving the tactile deformation that is happening at the surface.
Those are basically the nerve endings, aren't they, that have got specialised endings that can pick up things like being squeezed, stretched, vibrated.
Yeah, correct. So the fingertip contains thousands of these specialized nerve endings that responds to different aspects of any tactile interaction. There are some that responds more to stretch, others that respond to vibration. And overall, this small structure can encode any kind of stimulus happening on the skin surface with a very, very high resolution. So basically we can perceive tactile stimulus that are like very small, very tiny, with a lot of precision.
And can you actually see those nerve endings being stimulated? So can you see them deforming or stretching or effectively receiving the signals? Because obviously it's one thing to watch as the finger slides across the surface and these changes happening, but do you actually know that those particular nerve endings are responding to the stimuli you can see going in there?
So with our imaging technique, we cannot see those nerve endings responding, but we know from other studies in the literature that are collected with different techniques that those nerve endings respond to the sliding interaction. And we also know where these nerve endings sit because they sit precisely at the interface between the viable epidermis and the lower layer. And so we can infer from this that the mechanical stimulation we can image with our technique correspond to some precise nerve encoding.
And what does this reveal? When you do this, as you slide the digit across the surface and you see these changes effectively in real time, what's actually happening inside the fingertip?
The fingerprint ridges don't behave just like rigid bump that just bend sideways. Instead, what we saw is that the fingerprint ridges flatten and they shear internally. And this means that different part of the ridge that form like the fingerprint ridge slide and like they move relative to each other. And so under vertical pressure, the ridge structure compresses. And during sliding, there is like this shear that propagates to the internal layer of the skin.
Does it make a difference, though? Because if you look at a fingerprint pattern, how those ridges are organised, they're in sort of whorls or circles. So does that mean that certain directions of movement of the digit at certain positions are going to be interpreted differently? How does the nervous system overcome that?
Yeah, what was interesting to observe is that the deformations propagate through the internal layers of the skin, and there are like some kind of deformation that are enhanced by the skin layers. For example, we saw a high response during the stick to sleep transition. This means that when we start sliding something over the fingerprint, first there is a phase where there is no relative movement between the surface and the fingertip, and this phase is called stick. And then at some point, the fingertips start sliding over the surface. And when there is this transition between stick and sleep, there is the higher deformation into the skin layers. And this is because our nervous system is particularly interested into the transition between two different events. And this is what signal, for example, when an object is sliding from our hands, and we can respond promptly to the sliding of an object.
So fingerprints are not just for catching criminals. Giulia Corniani there. If you take a trip to Paris, inevitably you will go to see one of the world's most famous landmarks, the Tour Eiffel Tower, which was completed in 1889 by Gustave Eiffel to celebrate the Exposition Universelle, or World Fair, that was taking place in the city that same year. Eiffel added engravings to the first-level balcony on the tower of the names of 72 scientists who had achieved academic distinction during the previous century. But all of those named are men. Admittedly, there were far fewer women in science during that period, but there were nevertheless some standout individuals whose work has certainly shaped the world that we inhabit today. In fact, the Eiffel Tower itself relies on the material science know-how and calculations of one of them. And most of us are aware that Down's syndrome is linked to cells carrying an extra copy of chromosome 21, but almost no one knows who first counted them. Now, a new initiative is underway to engrave the names of 72 famous female scientists on the tower, alongside the men. CNRS Research Director Julie Batou has penned an essay for eLife showcasing the extraordinary stories of some of them.
The backstory is really not having women in science. For example, in the first floor of the Eiffel Tower, since the beginning, there is just male scientists. So 72 names. This underrepresentation is not that they were not there, they were not visible. The motivation is just to make these women scientists visible to the whole community and to the public.
And the scientists cited on the Eiffel Tower, these 72 names, who are they?
So there was a great scientist for this time, for the 19th century, in mathematics, physics, and as well biology. I don't want to give any name because we know them for a long time. I just want to focus on the woman that we propose. So 72 names of women, and for one which is Sophie Germain, because without her work as a mathematician, there will not be any Eiffel Tower. She put all the maths for the elasticity of the Eiffel Tower. She was not authorized to study mathematics. She used Monsieur Leblanc. She take a main name to have access to the education, and without her work on elasticity, the Eiffel Tower would not have been constructed. So Sophie Germain was really the woman, the scientist woman forgotten. And we want, the Association Pharmacians, we want her name to be back. So we propose her, as well as 71 other women, making really high contribution in physics, maths, and biology.
Are these all French researchers, or are these women internationally who've made their mark in history?
This is a very good question. Actually, there are women that make an important contribution to French science, but they are foreigner. For example, Rosalind Franklin, she was a UK person, but without the crystallography of the DNA, we will not get towards the structure of the DNA. And because she did all that in Paris, we propose as well her name.
So this list of 72 women who've made a mark on French science, pending approval, they are going to end up on the Eiffel Tower as well.
Exactly. We hope by the end of the year, so 2026, so that they can be engraved on the first floor on the Eiffel Tower, close to the main scientist, which is a wonderful, I think, sign for our society to put men and women. And these women, they make as well tremendous contribution to science, like mathematician, an engineer like Jeanne Guillot or Sébastien Guillot, which were amazing during the Second War and the First War to do a lot of work nationally, but as well internationally recognized.
You've written this really terrific essay for eLife in which you pick on 4 characters. Tell me why you chose the 4 that you did. I'm pleased to say I had heard of one of them, but the other 3 I hadn't heard of at all, and I was gobsmacked to learn that one of them discovered the extra chromosome in Down's syndrome, and I can't believe I never knew about her.
Yes, this is Marthe Gauthier, which as well is proposed for the Eiffel Tower. I picked them because of the PhD defense of Lisa Thoman. I did assist as an examinator to her PhD defense. And within the introduction, she really describes the work of Ethel Brown Harvey, her work on Hydra and on cell plasticity. The introduction was so well written. I say, Lisa, we have to write something on Ethel Brown Harvey because the Nobel Prize was given not on Ethel Brown, but to Hans Spemann, a man. The pioneering work was made by Ethel Brown and Ide Mangold, which was a PhD of, you will read the article, but so because I am an embryologist, we were to Ethel Brown Harvey, Ide Mangold, and then for me, Ida Henrietta Ide, which was the first woman engineer to get a PhD in Heidelberg. So for me, this is very important that people really know this woman. So Ethel Brown Harvey, Ide Mangold, and Ida Henrietta Ide, which for me were amazing in my field, but as well as the cytogenetics, Marthe Gauthier. Marthe Gauthier, she's discovered the extra chromosome of the Down syndrome, but this discovery was completely blacklisted by Jérôme Lejeune. Science is a collaborative work. It's not the work of one man or one woman. It's a work of a team. This was really what we wanted to say in this essay.
Julie Batu there. What a great story. That's where we have to leave it for this episode. You've been listening to the eLife podcast, which is produced by The Naked Scientist. There are previous editions, in fact, all of the previous 100 plus episodes of the programme, the references as well as full text transcripts, and how you can subscribe to this podcast over at nakedscientist.com/eLife. And The Naked Scientists also publish a weekly science programme that covers the latest leading science breakthroughs. You can find that at nakedscientist.com/podcasts. From me, Chris Smith, meanwhile, thank you very much for listening, and I look forward to your company in the next episode, number 103.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elifesciences.org.